Well, hello and good morning, good afternoon, and good evening, everyone that's joining us today. Thank you for joining our webinar on label-free imaging and nanoparticles in biological environments. I'm Carol Ray, Program Manager at CytoViva. I'll be monitoring the webinar this morning. During the webinar, please feel free to drop your questions in the chat option at the bottom of your screen, and we will address them at the end. I'll go ahead and introduce our panelists for today. We have our very wonderful Vice President at CytoViva, Byron Cheatham. I'll go ahead and turn it over to you now, Byron. Thank you.
Thanks, Carol, and good morning in the U.S., Friday morning, and Good Friday afternoon for participants in various areas of Europe. Thank you for joining us. We're going to spend some time talking about enhanced dark-field hyperspectral microscopy, and we're going to focus on how it's become a leading standard around the world for imaging nanoparticles in complex biological environments. The technology today is utilized in well over 400 research labs around the world, primarily that are involved in nanodrug delivery, nanobiosensor, nanotoxicology, and related applications for research. And what we're going to do is demonstrate in a very clear way with live samples today on the microscope the performance of the system, and then demonstrate how it worked with hyperspectral imaging to allow us to observe, confirm the presence, the location, and the relative amount of nanoparticles as they're interacting with a wide variety of biological matrices. We're going to talk about the principles of the instrument that allow that to work and how some of the patented optics and spectral imaging characteristics really produce this effect at a very high quality way. And then we're going to show some other examples of different types of applications in the space. So to get started, Carol, and everyone that's joining today, we're going to get out of the presentation mode here just for a second, and we're going to close that out, and we're going to see if our live preview screen for our live image opens. And yes, it is. So we actually have a live image this morning of nanoparticles in HeLa cells. And I'm going to put these in a little bit better focus here so that we can see it. So you should be able to see my screen. And so on the CytoViva Enhanced Darkfield Hyperspectral Microscope, we have a live sample. If I bump the table that it's on, you can see it vibrate a little bit, showing that it's a live sample. And so you can see in this image that we have some almost confluent HeLa cells all throughout this image. And these HeLa cells on an in vitro basis were exposed to 40 nanometer gold particles that had a peptide targeting protein on them so that they would be taken up by these HeLa cells. And so as we look around the sample here, you can see the cells, and these gold areas that you see here on the cells are actually the gold particles. So in about 30 seconds, I can plate this sample, I can focus it, and I'm instantly looking at 40 nanometer particles interacting, in this case, with mammalian cell culture, where a targeting protein has allowed the uptake of that. An important note is that the nanoparticles nor the cells have any special sample prep in order for us to be able to have this live observation this morning. It's completely label-free, which is a super important characteristic of that. If I want to observe nanoparticles, the last thing in the world that I want to do is to try to add clunky fluorescent labels to it that will affect the chemistry of the nanoparticle potentially. But even more importantly, if that fluorescent label in the context of the sample were to disassociate from the sample, I would just be looking with a fluorescent microscope at fluorescence that's disassociated from the nanoparticle. So it's really not a good way to try to look at nanoparticles, fluorescent labeling. With electron microscopy, the field of view would be 1/10 of what we're looking at right now at the most. And there are all sorts of really difficult sample preps that allow it not to be imaged in 30 seconds from the time you played it on the sample, but in a much longer time period. Now, electron microscopy has some significant advantages over anything optical, right? And so they can be very complementary. And that's not to say that it doesn't have a place, but for in your lab, on your benchtop, to be able to quickly and easily observe what's going on at the nanobio interface, this technique is the premier technique for doing that, and there and some advantages there. And also, as it relates to fluorescence, there may be a need to try to fluorescently label cells in some cases to look at co-localized information of nanoparticles in cell structure that's fluorescent. And that's a fantastic application for the system. And we're going to show some examples of that a little bit later on that take that into account. One of the things I want to do before I close out is I just want to do a digital zoom really quick to show you how detailed you can get with a dark field image, an enhanced dark field image of these cells. You can see now a little bit of movement as there's just vibration from lights and that sort of thing in the room, because we're at a 60x objective and we're probably another 20x magnified. We're about 1200x on this. What I want to do very quickly is just demonstrate for you with this 60x objective that has a very shallow focal plane, that as we focus in and out of the sample, you can see that some nanoparticles that are on different focal planes of these cells will actually come in and out of focus, just to sort of give you a sense of, in a very highly magnified way, mostly digital magnification with the camera software, we're able to see these nanoparticles and understand how they're interacting with these cells. One of the things to notice is that this is the nuclear membrane of this cell. Here we look at the nuclear membrane of this cell. And it looks like that the nanoparticles in this case are preferentially being taken up by the cell outside of the nucleus, which makes perfect sense given that their original size before they were internalized by the cell was approximately 40 nanometers, which is too big to cross the nuclear membrane of these cells under normal circumstances. But interestingly enough, it looks like that they're congregating around the nuclear membrane in some cases, which is typical of what we see. Now, it is possible to see nanoparticles, if they're small enough, that may cross the nuclear membrane. We won't go into that today for purposes of time, but that is something that's possible. So we wanted to show you first this live optical image, an enhanced dark field image of nanoparticles interacting, in this case, with these HeLa cells, just to give you a sense of the clarity and the speed at which that can be done. So with that, I'm going to close out this software. And now I'm going to bring in, of the exact same image, it's a slightly different field of view, a hyperspectral image that has been captured of this very same sample. Okay, so when we think about a hyperspectral image, it's a 3D image. Okay, and we're going to talk about what we mean when we say 3D. The three dimensions are, in the X and the Y, are the spatial dimensions. So from a visualization perspective, we're looking at a two-dimensional image of what are three-dimensional objects, the cells and the nanoparticles and that sort of thing. But we're on a specific z-axis focal plane that's consistent with the depth of field of the microscope objective that we're using, which in this case is a 60x. Here we have the full field of this hyperspectral image. Now, we talked about that it's a three-dimensional image, okay? And so the third dimension of this image is not spatial, it's spectral. So you see, as I start to zoom up these pixels, and it gets very pixelated in this zoom image that we're looking at here, we can see the pixels. And so the third dimension is spectral, not spatial. X and Y spatial, Z is spectral, meaning that behind every pixel is all of the spectral data for that pixel's spatial area from 400 nanometers to 1000 nanometers. And to demonstrate this, I'm going to right click in this box. So here is the Z profile, its spectrum. And I'm on a tissue area here, a cell structure area here. And as you see, as I move around, you see that there's some change in the actual spectral profile. There's slight nuanced differences in this cell from pixel area to pixel area. And just for reference, the pixel sizes here are about 240 nanometers, 0.24 microns in terms of size. Now, we talked about these nanoparticles being originally about 40 nanometers. There's a couple of things to consider here. We're not looking at the nanoparticle. We're looking at scatter from the nanoparticle, from the surface plasmon resonance. And we're not looking, we're also looking at the me scatter from the cell structure here. And we're going to talk about how we create this image and how it's the mechanics behind it and the engineering behind it in just a second. But first, I just want to show the performance of the system so you can get a sense of it. But if I go into the spectral profile plot and I want to collect spectrum, I have spectrum of tissue here. Now let's go in and collect some spectrum of the gold nanoparticles. The other thing about this gold is that once it's in the cell, it's absolutely going to aggregate. Not only are we looking at the scattering of the nanoparticles, but we're looking at the scattering of aggregates of the nanoparticles, and that's completely predictable. Endosomes in the cell are the garbage collectors, and boy, these nanoparticles are going to get their attention. And so a lot of times what we're looking at when we look at a large aggregate, maybe such as this, is that we're looking at actual endosomal uptake maybe of these nanoparticles in the cell. I'm not a cell biologist, so I'm not going to try to give you a cell biology class on what's really happening. Most of you, or many of you, could probably give me one, and no doubt. I'm more of a microscopist than a spectroscopist. But this gives you a sense of how you can see in spatial context the cells, the nanoparticles interacting with the cells, and measure the spectral difference at the pixel level between, for example, what appears to be the nanoparticles in the cell and the cell structure itself. If I have all of the spectral data, I have the ability to conduct detailed spectral analysis to help me make some confirming aspects of the data that we're looking at here. So, for example, one of the things that we can do is we can do spectral mapping. So if I look here and I want to, I can do spectral mapping of spectral libraries. And so here with mapping methods, we can use the spectral angle mapper classification, for example. And I've already created the spectral map of the nanoparticles for convenience of time for this particular sample. And I'm going to apply that map. And I do that here by just simply clicking on the interactive class tool that provides that data. And what it's actually doing is that it's demonstrating, and you can see here in this zoom when I cut it off, it's demonstrating everywhere that nanoparticles are present. And we built this with a spectral library of these nanoparticles in this cell structure, and we filtered that spectral library against the negative control. Again, in the software, we have the ability to do all of these capabilities quite rapidly and with a high degree of accuracy. So in this image, not only can I understand that I have nanoparticles interacting with cells, I get a sense of where and how those nanoparticles are interacting with cells. And I get a quantitative class distribution mapping of these in a two-dimensional area. And I can then report that data here. Let's see if I can pull this over. Just one second here. So this is the actual class distribution data that comes from this mapping, okay? And it says that 4% of all of the pixels in the image, in this two-dimensional spatial image with the third dimension being spectrum, are matching and mapping for the presence of the gold nanoparticles in the cell, okay? And so that's just demonstrating that this morning. And so wanted to actually here in the first 15 minutes of the webinar this morning, show you some live data that came right off of the microscope that I'm sitting here. You can see a microscope behind me. It's in our imaging lab here at CytoViva. That microscope is identical to the one that I'm using here this morning to be able to demonstrate this. And with that, I want to actually move all of this data out of the way and transition here in the webinar to be able to show how we actually get this data here. So let me pull this out of the way and now go back to the presentation, open it up. Let's see here. Apologize. Okay, so we have the presentation. Let's go through here and let's talk about what the technology can do in terms of its major functionalization. It allows the ability to observe nanoscale elements that are interacting with either labeled, fluorescently labeled, or unlabeled biological or material matrices. Let's take a look at another sample that's very relevant. We know that nanoparticles, specifically silver nanoparticles, are being developed as antimicrobial agents, and they have incredible properties in that regard. And there are many peer-reviewed publications that demonstrate this, and there are many that publish using data with CytoViva on this. And in this example, we're looking at a bacteria, Bacillus subtilis bacteria, that's been exposed to silver nanoparticles. And in this circle here, you can see this silver nanoparticle. It appears blue because the silver nanoparticle has a surface plasmon resonance at this size that's in the blue range. And if I look at this dark blue spectrum here, that's representative of the silver nanoparticle with a peak at about 470, 480 nanometers, which is in the late blue range along the electromagnetic spectrum. If I look at the bacteria membrane itself, here circled in white, and then I represent that with this white curve here, we see that there's a tremendous redshift, a tremendous change in the bacteria and the bacterial spectrum, right? However, where we look at where the silver nanoparticle appears to be interacting with the bacteria on a qualitative observational basis, we can make some quantitative conclusions about that based on the pixel level spectrum of that silver nanoparticle that appears to interact with the bacteria. And we can confirm that because now if we look at the light blue spectral curve, it's very different from the silver nanoparticle isolated. It now appears to be a hybrid spectral curve of the silver nanoparticle and the bacteria, because that's in effect what it is. The biocorona of the bacteria is affecting the optical spectrum of the silver nanoparticle and allowing us to confirm that that silver nanoparticle is indeed interacting in some way with the bacteria. And of course, if it's the active agent, the fact that it's interacting with the bacteria can provide really good information. There have been a number of papers published, one in particular that I can think of at IIT Madras, where the group there looked at silver nanoparticle bacteria interaction and was able to do things like demonstrate lysing of bacterial membranes, individual bacterial membranes, based on this interaction and some other things. So we can measure the pixel level Rayleigh scatter, which would be the case of the silver nanoparticle, or Mie scatter, which would be the case of the bacteria itself. But we can measure the optical spectrum of anything. If something is producing photoluminescence, we're going to look at an example of phosphor dye in a second in a biosensor application. That phosphor dye produces emission. A quantum dot is going to produce a very distinct emission, or more traditional fluorophores that are utilized for fluorescence imaging. All of those, as well as scatter, whether it's surface plasmon resonance scatter or other types of scatter, can be measured by this technique, and they can be sorted and unmixed with this technique with a high degree of accuracy, which allows us to do spectral mapping and other analysis, not only in cells, such as the bacteria or such as the mammalian cells that we were looking at, but also in tissue and other environments. Even in environments where It may be a pure material science application. If it were on a silica substrate, for example, and you were looking at perovskite nanoparticles on a silica substrate, you could measure that as well, for example. The microscope system that's sitting beside me here in my lab this morning is identical to the one that you see on the screen. And this system looks like a research-grade optical microscope, and it looks like a pretty simple one. And in fact, that's purposely what CytoViva wanted to create, was a very basic-looking research-grade optical microscope, because it's easy for us to teach almost anyone to utilize this technique when we use a very simple research-grade optical microscope that has the components integrated onto it that allow for the very powerful image, hyperspectral data, hyperspectral analysis capability that you've already seen, that we've shown this morning. Now, it's important to note, and we're going to talk a lot about enhanced dark-field optics, transmission enhanced dark-field optics, that are designed to allow us to see nanoparticles, plasmonic nanoparticles as small as 10 or 20 nanometers, metal oxides in the 20 to 30 nanometer range, even lipid nanoparticles in the 70 to 80 nanometer range. And also there was recently a paper published looking at viruses, coronaviruses, for example, and coronavirus uptake in mammalian cells, which were about 90 to 100 nanometers, and the ability to even observe the scatter there. Not all nanomaterials scatter light equally based on their chemistry. So different types of nanoparticles are going to scatter light differently, and so the detection limits would be different. And we can talk with each of you about questions that you might have about specific types of nanoparticles and what the detection limit might be in this technique. But what we want to show here is that we take our basic research grade optical microscope, we install patented enhanced dark field optics in the condenser mount of the microscope. We have some fluorescent capabilities. We use a translational stage to do a very high resolution hyperspectral image capture. We'll talk about how that works. And on the dual port camera mount of the microscope, we have two systems here. The first system is the spectrograph and SCMOS camera that are integrated together to create the spectrophotometer that creates the image that has the spectral data in every pixel. Behind the dual port here, we have a standard research grade optical camera. That's what I used to show you the optical image that we first looked at of the cells and the nanoparticles. And you can simply switch back and forth between the two. You can, even if you have dynamic processes going on with your sample, with the optical camera, you have the ability to capture that as a video if you want, which can be very valuable. However, the microscope can be equipped to do anything that a microscope can typically do. It can operate with transmitted bright field. In the case of stain pathology samples, that might be the best imaging modality. The system can support that and can support looking at nuanced changes in stains with H&E stain pathology samples, for example. Transmitted enhanced dark field microscopy is the primary focus for the microscope technique when we're looking at the nanobio interface. And that's what we were using with all the samples that we showed you already this morning. We can work in reflection mode as well. And this microscope is equipped with reflection optics. Let's say, for example, that you were in more of a pure material science application and you wanted the ability to look at quantum dots, I mentioned that on a silica substrate, or you want to look at something else on an opaque substrate. You can do that in reflection mode. Or if you're looking at something that's on a very dense tissue in a biological format and light can't get through it in a transmitted light mode, you can go in reflection mode and do that as well. You can do reflection bright field or reflection dark field, depending on the application. And then you can do traditional fluorescence microscopy, or CytoViva has a very interesting technique that we refer to as dual mode fluorescence. And I'm not going to spend a lot of time on it today, but dual mode fluorescence, and this is the dual mode fluorescent module and the light source that's optimized for that, allows the researcher to see unlabeled nanoparticles or unlabeled sample elements and fluorescently labeled sample elements in real time with no overlay. and capture hyperspectral images of that data as well if you want to. And so the application for that could be, for example, that I want to understand how nanoparticles are interacting with mitochondria in cells. And I put a GFP mitotracker on the mitochondria in the cell. And I want to see how that green mitotracker mitochondria label is interacting with the nanoparticles, or the nanoparticles co-localized with the mitotracker green, which is consistent with the mitochondria, yes or no. That kind of information can also be gathered from this. And the system can operate in the visible near infrared or the shortwave infrared. We'll talk about the different wavelengths for that and what applications may fit for which. Before we go deep into the technology, I just want to show you some examples, though, of the different types of research institutes around the world that utilize the technology, and more importantly, the types of research applications that they're involved in. And you see a lot of applications related to nanotoxicology. You see nanomedicine and nanodrug delivery related applications, such as what we have here. And also a lot of just more traditional nanomaterial synthesis, where researchers are developing the nanomaterials themselves and need to be able to measure in real time and quickly the optical spectral response, and need to understand aggregation versus dispersion and homogeneity of nanoparticles. That's another application of what you can do with the system. As we referenced, the system is being deployed in ever-growing numbers around the world. There are over 400 of these systems, primarily in nanomaterial research labs around the world. Almost 1,700 publications now you would find if you went out and did a Google Scholar on CytoViva, where CytoViva has been referenced in these publications. I want to very quickly just take you through some examples to sort of show some of these publications. And everything I show is going to be what I'll refer to as a methods paper, looking at hyperspectral microscopy, or Darkfield hyperspectral microscopy, as the method in the nano or nanobio world. And this first was done a number of years ago by Enrique Toma in Sao Paulo, at the University of Sao Paulo. And here they were looking at different sizes and different shapes, nanoparticles, gold nanoparticles in this case, to demonstrate how they could confirm different sizes and different shapes of nanoparticles in a mixed sample utilizing this instrument, because different sizes and different shapes of plasmonic nanoparticles create different optical spectrum. And that can be characterized, and then you can utilize that characterization to be able to delineate between those different types of plasmonic nanoparticles in a mixed sample, for example. And so this is a very relevant early paper that was done. This paper by Dr. T. Pradeep and his team at IIT Madras and Kamalesh Chaudhary is a really interesting paper, a Nature paper, where they were able to demonstrate, along with the use of some polarization in some cases, a very small, a 10 nanometer by 30 nanometer aspect ratio gold nanoparticle, and able to track a single gold nanoparticle in and out of the cell. I would recommend that you go read this paper and to understand everything they did. It's a very dynamic paper. And shows the power of the instrument, but also showed the novel ingenuity of this lab to utilize the instrument to demonstrate some very cool single nanoparticle cell type work, live cell type work. They actually tracked this single nanoparticle in and out of a live cell. Very interesting stuff. And then finally, this paper is more of a general methods paper. Paula Zamora Perez was the first name author on this paper, and she actually led the publication of this work and paper as a graduate student. But it's a very interesting paper that looks at how a lot of different types of nanoparticles can be effectively studied in different cell and tissue environments with the technique. Again, another methods paper. And then finally, Dr. Sarah Brenner at SUNY Albany and the Nanoscale Nanofab Center there in Albany in New York State published a paper looking at metal oxide nanoparticles, in this case focusing exclusively on tissue. And this is a human exposure nanotoxicology related study, but just wanted to take you through some examples to give you some very specific applications for how the technology and the system is utilized. Now I want to transition and talk about how we get these images and how we get this hyperspectral data and the underlying technology that allows us to do that. And we want to start with what I consider to be the most important underlying technology, and that's our patented enhanced dark-field microscope optics. This is a picture that demonstrates this. This enhanced dark-field illuminator replaces the standard condenser in the condenser mount of the microscope underneath the stage of the microscope. And this system is designed to maximize signal to noise ratio. If I think about signal from the scatter of the nanoparticle versus noise from stray light that comes from the light source, okay? And I'm not going to spend a lot of time on the physics on this. I'm just going to take you through more of the mechanics of this. But this system is designed to minimize light loss and take the light from the light source. And in most cases with nanomaterials, especially when no fluorescence is involved, we're using a very simple quartz halogen light source that has an aluminum housing around the bulb reflector, so that we can push near infrared wavelengths, so we can go from 400 nanometers out all the way to almost 2 microns in terms of the excitation. But that excitation is very smooth, because if I'm going to capture a hyperspectral image, I don't want my source illumination to have lots of spikes in it. I want it to be very smooth, so that when that light interacts with the objects of interest in the sample, that I'm able to see that more of that pure reflectance spectrum from that guy. Now, we still need to do some normalizations to remove instrument response and that sort of thing from the sample in the hyperspectral image oftentimes, but you don't want to try to remove spiky spectrum that you would get with LED or that you would get with mercury vapor and other types of light systems. If you do need to use those, though, for example, you can use notch filters and other things to try to minimize that. Now, with the dark field optics, we're using lenses and mirrors in the dark field system, and what we're doing is we're trying to create what I'll call perfect color illumination. Many of you might be familiar with color illumination and the strict technical definition of that, or maybe the simple definition of it. Color illumination is to focus the light from the source light onto the condenser. And so this is actually the condenser mechanism of the enhanced dark field illuminator. But this system does something very unique. It changes the geometry of the light to very precisely match the geometry of the condenser. And that's one of the key elements that allows us to improve signal to noise. Because if I don't have stray photons floating through the system here, I don't have stray photons in my image. I get a very narrow oblique angle illumination. Dark field is oblique angle illumination, 65 to 75 degree angle illumination, so that it comes at this shallow angle and the light interacts with the sample. Scatter from the sample goes into the objective, but the source light, the angle of the source light is steeper than the angle of acceptance, or more shallow than the angle of acceptance of the objective. So I don't get source light, and that's sort of the technical definition of dark field. And to the extent that I'm able to minimize source light and maximize scatter, I get this significant increase. And I want to show you what that increase looks like. And I'm going to show you through a paper that was published by a research group in Korea. And it was a research group that for a number of years had used CytoViva's dark field optics, enhanced dark field optics, to do microchip electrophoresis work, where they're looking at iron oxide nanoparticles, native single superparamagnetic nanoparticles. It's iron oxide. And so in this image here below, they're showing images of the iron oxide nanoparticle utilizing CytoViva's enhanced Darkfield. Now, they also had a standard, I think it was Olympus Darkfield optics that were sitting in their drawer, and they wanted to just do a simple comparison to see what happens. And when they look at this, you can see there's a nanoparticle with the Darkfield optics with Olympus. But if you'll notice, the signal is very weak and the noise is very strong, which is inconsistent with what you're trying to do when you're trying to look at nanoparticles optically. And you see here, you have a very strong signal from the nanoparticle, and you have very little noise. And when they measured the signal to noise ratio difference, they measured across multiple different samples that they looked at about a 10x increase with the CytoViva Darkfield optics. These optics make it easier to see smaller materials, and it makes it easier to get the sample. I can be looking at nanoparticles, 20, 30 nanometer objects in almost any environment from the time I plate them to I get them on the microscope in less than 30 seconds. That's the key. It's really easy. We can teach someone who's never used an optical microscope how to do that in just a few minutes. It's not very hard to do at all. And that's very helpful. If you want something sitting on your benchtop, that will allow you to be able to observe in real time as you're modifying and changing the optical or the research parameters of your samples as you're working on them. I'm going to transition now and talk a little bit about hyperspectral imaging and how it works for those of you that might not be very familiar with it. So in hyperspectral imaging, as you saw, every pixel of the image that we looked at earlier had the optical spectrum in it, right? And so we can capture optical spectrum in the visible near infrared or the shortwave infrared. In the visible near infrared, we're operating from 400 to 1,000 nanometers. And I would say that for 90+% of the engineered nanomaterial type work that we are involved in, that's typically what we're looking at is working in the visible near infrared range. But sometimes we work with researchers that may be developing nanoparticles that will get away from tissue autofluorescence, for example. And in that case, we can also operate in the shortwave infrared range. We can collect that spectrum from 900 to 1700 nanometers, for example. So we can suit a wide range of needs. One of the things that we would do if we were working with any nano group is to understand their samples, understand which ones would work better in which wavelengths, and make those kind of recommendations. So the pixel sizes that we were looking at, as I mentioned earlier, in the image we were looking at, were about 240 nanometers or so. Pixel sizes get down to about 128 nanometers if we were at, say, 100x versus 60x with the microscope. An important note about hyperspectral imaging is, especially CytoViva's hyperspectral imaging, is that it can produce very high spectral resolution. And that can be very important because in basic research, you don't really know what spectral resolution you need, right? Let's say, for example, that you wanted to try to measure a functional group on a nanoparticle, and that functional group was creating just a very nuanced change in the optical spectrum of the nanoparticle. Having 2 nanometers of spectral resolution would help you there. Let's say, for example, that it created absorption between 460 and 470. So with 2 nanometers of spectral resolution, that spectral change is going to be easily recorded and easy to understand that it's not an artifact. It's truly a function of, in this case, the functionalization of your nanoparticle that you're looking at. But the coolest thing to me with regard to hyperspectral images is that it's images. The hyperspectral data is presented in spatial context. It looks almost identical to the optical image, either from the optical camera or from your eyes on the ocular of the microscope eyepiece. So you get all of the spectral data from your sample at the nanoscale, and you get it in full spatial context of your sample. And that can be very valuable for helping understand, for example, how nanoparticles are interacting with a wide range of matrices, because you get the spatial context of the matrix that you're looking at, the nanoparticles, and how they're actually working together. So, and if I have all of that data, I can perform lots of detailed quantitative analysis, and the mapping and the class distribution that we showed you earlier are an example of that. Here is the power of proper dark field optics for hyperspectral imaging. The exact same nanoparticle and the exact same pixel from that nanoparticle through an automated stage that has very high degree registration. The exact same setting on the hyperspectral camera. The only difference is using standard dark field and CytoViva's patented enhanced dark field that create the very high signal to noise ratio. Signal to noise in spectroscopy is very important, and it doesn't matter what spectroscopy it is. And of course, it's important in hyperspectral imaging, as you can see here. So at the nanoscale, good dark field optics can mean the difference between reliably good spectral data or not. And it was always the chicken or the egg. Once we created the dark field, we knew we had the optics to really provide lots of incredible spectral data at the nanoscale. And that was what led to the hyperspectral imaging, was knowing that we had this really great high signal to noise imaging that we could apply to hyperspectral imaging at the nanoscale. I want to transition now away from the technology, and I've shown some examples, but I just want to show as we close a few more examples of how nanomaterials can interact in biological matrices, and also an example of what we can do with regard to nanomaterial characterization itself. And I want to start out with a nanomaterial characterization sample. These are silver nanoparticles. And these silver nanoparticles are represented to have a mean diameter of about 30 nanometers. Now, it's important to note that in the technique with dark field, we're not looking at the particle. We're looking at the scatter of the particle. And the scatter of the particle can be orders of magnitude larger than the particle. That's why we can see it so easily with an optical microscope. That's the whole point and purpose behind dark field optics. And here we're looking at 30 nanometer silver nanoparticles that are in very different environments. These silver nanoparticles, they're the same particles, are in water. And in that environment, we see that there's aggregation. And as the silver nanoparticles aggregate, because of their surface plasmon resonance, they redshift. So we see them change as they're smaller, they're blue, they go to green, they go to yellow, they go to red. But also in the water, we're seeing oxidation, H2O. The oxygen in the water on these particles is going to cause oxidation, and that's going to affect the optical spectrum to a certain degree of these nanoparticles as well. And so we see what seems to be a very heterogeneous sample set here of these nanoparticles. And if we look at individual nanoparticle spectrum that are measured here, that's validated in a more quantitative manner, where we see very heterogeneous particles. As they redshift, you'll notice that we get a peak intensity that rapidly changes from the ones that are blue shifted, right? Because an aggregate of nanoparticles is going to produce more relative intensity of its peak as opposed to nanoparticles that are not. Now, if we come here at the bottom, these are the very same nanoparticles that are put in an ionic solution that are designed to stabilize the nanoparticles and keep them dispersed. And so just from a qualitative observation perspective, with CytoViva's dark field optics, you can see how these guys are very homogeneous in terms of the way they look. But if we measure single pixel or single nanoparticle optical spectrum, we're validating in a quantitative way that heterogeneity, that homogeneity that we see in the nanoparticles. And so we're validating with data what our eyes are telling us from a quantitative perspective. And this is an example of looking at how nanoparticles in a different environment will react to the different environment in measuring that in a quantitative manner. Now, we showed you gold nanoparticles in cells earlier. I only want to show this example to show the stark difference between gold nanoparticles that have been internalized by these cells versus the very same cell structure that are negative controls without nanoparticles. And so it's intuitively obvious to the most casual observer that there's something very different about these cells from these cells. And of course, that's the presence of the gold particles, which are the only variable changed in the experiment. However, we still want to quantify that. We want to quantify that with the optical spectrum and the optical mapping and the class distribution which gives us information about the relative amount of nanoparticles that are there in the two-dimensional image. We're just now going to bump through some other different examples here very quickly of nanoparticles to look as we come up on the hour. These are iron oxide nanoparticles that are in cells, and they've aggregated greatly in these cells. These white objects that we see here are the silver nanoparticles that are in the cell structure. And here's, again, a negative control. And there's also a process, and I'm not going to go into it, where we can build spectral libraries of things like nanoparticles, filter them against negative controls to ensure that the spectral library for the nanoparticle target maps without any false positives. And there's a process for doing that. There's a special analytical tool in the software called Filter Spectral Library. The kinds of things that you would learn if you had a system in your lab. Now, finally, we want to transition to some tissue and demonstrate some applications in tissue, looking at silica, inhaled silica in mouse tissue. And so this is an example of where the live mouse inhaled the silica nanoparticles. The mouse was sacrificed, and then sectioning of the lung tissue occurred. And you see the lung tissue here. And then we see these white punctate areas. If we look at some of the chosen spectrum here, notice that the tissue spectrum looks very flat. Because the tissue spectrum was very homogeneous, we normalized this sample against the tissue spectrum, against tissue control. So the tissue spectrum itself produces this very flat spectrum, but anywhere that we have nanoparticles, in this case silver nanoparticles, we see a very different optical spectrum that allows us to map that. And here's the spectral library of the silica nanoparticles that we used to map. And again, this spectral library was filtered against the negative control tissue to make sure that the only thing we're mapping with this spectral library are the silica nanoparticles that had been inhaled by the mouse in this case. Now we want to move on and just look at some multi-wall carbon nanotubes, but we want to look at that in stained tissue. As many of you would know that have ever worked with a pathologist, their comfort zone with any tissue is to make sure that they have that stained. That's their roadmap for the tissue. And you can also look at nanomaterials in stained tissue. Here we're looking at multi-wall carbon nanotubes and the scatter from these nanotubes. I've often heard people say it looks like when they're dispersed like this, spaghetti in the tissue, and indeed it does. And you can see these two here in the tissue. This is the representative spectrum of the multi-wall carbon nanotubes, representative spectrum of the stained tissue. And again, you see this incredible increase in the amplitude of the signal from the carbon nanotubes because they're highly efficient scatterers of light relative to the tissue. We could normalize these to an intensity factor of 1 so you could compare them a little bit better, but I wanted to show how the reflectance intensity is so much stronger in the case of these nanoparticles versus the tissue. And now I want to transition and show just one more example that shows the power of being able to characterize functional groups on nanoparticles. And here we're going to look at polymer nanoparticles, again, a different kind of nanomaterial, okay? And these polymer nanoparticles are just bare in solution. The very same polymer nanoparticle has now had a small molecule drug, an API, an active pharmaceutical ingredient, cisplatin, which is a well-known chemotherapy drug that can be utilized for chemotherapy treatments. Cisplatin can have harmful effects on a wide range of tissue, including cancer tissue. To the extent that I'm able to create a functional group for something like a polymer nanoparticle to target cancer tissue, and then I can also cleave the cisplatin onto the surface of that polymer nanoparticle, I'm able to target the cisplatin small molecule drug to the nanoparticle and have it specifically target the tissue or cell area. But what I need to do is understand how well first I'm able to functionalize, in this case, the polymer nanoparticle with the cisplatin. And one of the ways that I can confirm the efficacy of the functionalization is with Darkfield Hyperspectral Microscopy at the individual nanoparticle level. We see here that the cisplatin, even though it doesn't really change the way that the nanoparticle looks with your eye, it's dramatically changing the way that that polymer nanoparticle is absorbing and reflecting light. And you can see here where we're measuring that change. Where there's a reflection peak, now there's an absorption dip. Dramatically different changes in the spectrum as a result of the presence of the small molecule drug. I can't tell you that I understand the chemistry of the small molecule drug to explain exactly why that's happened. But cisplatin has as its part of that is platinum, right? In the chemistry of the drug, I believe, and so that's partially responsible for that. Now, interestingly enough, these nanoparticles are three-dimensional spheres, and the hyperspectral image, from a spatial standpoint, is a two-dimensional area of the sphere. So the particle sort of appears flat, but we know it's a sphere. But because of where we're focused on it, the cisplatin is on the outside of the particle. So if you'll notice, everywhere that we're spectrally mapping the cisplatin, it's on the outside. Okay, it's not on the inside of the particle per se. And we also can clearly see, just from these very small number of particles that we're looking at, is that this group's done an excellent job of functionalizing what appears to be each particle, in this field of view at least, with the cisplatin, which gives them some great insight onto the efficacy of their effort to functionalize these nanoparticles, in this case with the small molecule drug. Wanted to show that as an example of that. And then the last example that I want to show is a really cool, it shows the power of hyperspectral imaging, looking at two distinctly different sample elements in a single sample. We're looking at photoluminescent dye, that's in the green range, and plasmonic nanoparticles that have been tuned based on their size, I imagine, to produce reflectance spectrum in the near infrared. This is looking at the sample. So this is a biosensor application, as I understand it, and it's a hydrogel that has polymer capsules. Some capsules are loaded with the phosphor dye that photoluminesces. Some capsules are loaded with the gold nanoparticles that produce this near infrared spectrum. And so each one of these two different polymer capsules, they sort of activate based on different interacting with different biological chemistry. And I can't provide any of the details on that beyond that's the way that I understand that they were created. What's important, though, is that when you're looking at this hydrogel with these polymer capsules, is to understand how many are loaded with phosphor dye, how many are loaded with gold nanoparticles. And while you can kind of get a sense, even in the dark field mode, that you can see some green here, maybe some green here, and some gold looking here, but it's purely qualitative, and it's a little fuzzy, quite honestly, because we're looking at these polymer capsules in this thick hydrogel, for example. But applying hyperspectral imaging analysis capabilities to this, I can map every capsule that's loaded with phosphor dye. I can map every capsule that's loaded with gold nanoparticles. I can get a class distribution that can tell me that in this image, about 13% of the area had gold nanoparticles and about 4% had phosphor dye. Now, this is a really small field of view. You would need to look at more fields of view to really get a good statistical breakdown of this. But if I was trying to go 50-50 with this, I'd know that I had an imbalance. It's more than 2-to-1 polymer capsules with gold versus phosphor in this application. It's not statistically relevant, of course, but the idea here is to show that we can do that. Now, they chose phosphor dye and gold nanoparticles tuned in the near infrared, specifically because they knew that they would produce optical spectrum that are far apart from each other and allow them to do this test very easily and very accurately. So this is mean spectrum, just showing the difference between the phosphor dye, and it's green because it reflects mostly in the green. It has absorption in the red. The near infrared nanoparticles have their peak that's primarily over here past 800 nanometers, which is in the near infrared range. Okay, so this is an application where hyperspectral imaging can give you the spectrum of photoluminescence emission and surface plasmon scattering in the same image, and record that at the same time in a single pass. And it speaks to the power of optical spectral imaging and its ability to do that, which is quite unique and can have lots of applications and utility. So that's the end of the formal part of this presentation. And so, of course, you see our website there. If you want to learn more about the technique, info@cytoviva.com will come into the main email to the company here if you have specific questions that you might want to ask. And Carol, I see that we have some questions and some things in the chat. And if you don't mind, I'll ask you to manage the questions and let's maybe start answering or asking the questions, and we'll do our best to answer them and to the extent we can. And if we can't, we'll get back to you later on them, in fact.
Absolutely. We had some really great questions. So here we go. So the first one is from Madhu. Is it possible to record a video in Darkfield/Brightfield in the CytoViva Horiba integrated enhanced Darkfield Hyperspectral Raman microscope? If yes, then how?
Well, as long as you've got an optical camera that's integrated with that, Madhu, you should be able to do that. Absolutely. There shouldn't be a reason why you couldn't, is the short answer. It just would be a matter of making sure that we had an optical camera that was video capable as part of that system. That should absolutely be possible.
Thank you. Our next one is from Andreas. Is the spectral profile normalized by the spectrum of the light source? Why do most signals have a close to zero value near 400 nanometers?
Andreas, it's a great question. So typically when we normalize the data, it's typically normalized against the source illumination, right? And so, and I call it lamp subtraction, but I get in trouble because the math is not subtraction, it's division. But yes, that's the process that's typically used. However, not always. For example, we were working with a group this week that works on silica substrates with a film on it. And in that scenario, the substrate with the film is very homogeneous, and you could normalize against that substrate as well. So it's not just exclusive to the lamp. It can be in the silica and tissue example, we normalized against the tissue. So there's a couple of options that you can use there, but most typically it is against the lamp. Now, in the examples that we showed, there weren't much signal at 400. The light guide that we were using had a cut-on at 420 nanometers. And that's why we could have used a light guide that started at below 400 if we wanted to. This light guide that cuts on at 420 goes all the way out past 1,000 nanometers. And so generally with these kinds of samples, we're looking at more data out past, say, 800 nanometers. And the light guide that cuts on below 400 it gets very weak out past about 700 or 800 nanometers. So that's why we didn't see much signal there.
All right, next, can the spectral profiles be predicted through analytical models, such as weak phase objects and Rayleigh scattering?
It can. I've seen people do that math to do that. I've even seen people take the optical spectrum of something and conduct the math to know what the wavelength number is for the Raman signal of that, for a known signal. So yes, there's a lot of analytical analysis you can do with that signal if you understand the math. I will admit that's above my capability to be able to do, but I've seen some really smart customers that do similar work to that.
Thank you. Next, what do the nanoparticles bind to in the cell? Well,
so and again, I'm not a cell biologist, and I'm not going to try to pretend that I'm one here today this morning. But I've spent a lot of time in cell biology labs and where nanoparticles are being utilized. And what typically happens is that the nanoparticles, well, they don't have to have a targeting peptide on them. If I had macrophages, for example, macrophages are cells that are designed in the blood system to seek out foreign matter and to eat it, to endocytose it, for example. And so in that case, nanoparticles that come in contact with a macrophage cell are going to get endocytosed by the cell and are going to be internalized by that cell. But also, if I'm looking at, for example, trying to target nanoparticles to specific cells, there are targeting proteins and peptides that will allow a nanoparticle to be preferentially endocytosed or absorbed by a specific type of cell. And then sometimes that nanoparticle, depending on other functional groups, can go to specific areas of the cell, as I understand it. And the chemistry in that's, again, way above my pay grade. Now, once it's inside a mammalian cell, most typically the endosomes are looking to try to scavenge and clean that up. But I do know that we have had researchers, Dr. Devika Chithrani at the University of Victoria comes to mind, where she created very small nanoparticles in about the 12 nanometer range. And she had a targeting protein that allowed those nanoparticles to cross the nuclear membrane, which is quite a feat. But she's published numerous papers demonstrating that. So depending on how you functionalize the nanoparticles, their size and other characteristics, they can be taken up by the nanoparticle and they can co-localize in some very interesting places in the cell. And it never ceases to amaze me what these researchers can do with that.
Absolutely. All right. Next is from Abner. Are we able to confirm with the Z spectrum that the particles are inside the cell and not around it, that they are not attached to the plasma membrane, for example?
Again, so the biocorona of the cell can really help make that determination. If you'll notice, even at 60X, when I was focusing up and down in the cell, you could see nanoparticles appearing and disappearing, right? And so you can just get, from a qualitative perspective, you can understand that, and you could actually capture a video of that if you wanted to. CytoViva also produces a 3D module, and I didn't mention that this morning, and it's a very important aspect of that, where we can capture multiple two-dimensional slices through a cell. And then we can conduct deconvolution with some very specific patented deconvolution algorithms that we have, and put those back together to allow us to understand where the nanoparticle is in the X, Y, and Z focal plane. And the 3D capability is another capability we have that provides that in a more quantitative manner. But also there have been papers published. Dr. Rebecca Drezik's lab at Rice University was one of the first that comes to mind, where she was able to demonstrate that the biocorona effect of the cell is different on the nanoparticle in different areas of the cell. Now, this was a highly complex methods paper that she produced, but it demonstrated that capability to know that they're not only not on the membrane, but inside the cell, but in different subcompartments of the cell. And Dr. Chithrani, who I mentioned earlier at University of Victoria, was able to demonstrate that when a nanoparticle was inside a cell and it crossed the nuclear membrane, there was again a change in the biocorona of that nanoparticle in the nucleus versus outside of the nucleus that resulted in a shift in the spectrum. This is very detailed work, and this work that was conducted over not days and probably not months, but years. But that sort of ability is there with the system. But honestly, the easiest way is if you have the 3D capability with the CytoViva system, and you could do 3D imaging to know where nanoparticles are in the X, the Y, and the Z.
Thank you. All right, next, can spectral libraries be imported/exported into the software for pattern recognition purposes?
Well, one of the things that you can do, so we're using a highly customized version of hyperspectral image analysis software called ENVI. But you can capture hyperspectral images with this. You can port them over into MATLAB. You can use Python, anything that will allow you to do pattern recognition, artificial intelligence, whatever you want to do, you can use this data in these hyperspectral images to conduct any sort of spectral analysis that you want to do. And that can be supervised algorithms, unsupervised algorithms, principal component analysis. Again, this is very rich data sets, and it's being utilized for a wide range of applications. For example, there's some pathology applications where some very specific algorithms are being created to do some very unique things in pathology. And there's some other applications in material science where that would apply to as well.
Mm-hmm. All right, next from Soha, two questions. So the first one, can we observe and measure the carbon dots with respect to the antibiotic resistance of the pathogenic clinical isolate at hand?
Wow, that's a deep question.
It is.
So the carbon dots, I don't know, typically carbon dots are designed, as I understand it, that they can have unique spectrum based on how they're designed. But I'm not sure about these. But with respect to the antibiotic resistance of the pathogenic clinical isolated hand, you can understand how the carbon dots are interacting with the pathogen. And Dr. T. Pradeep at IIT Madras has published papers looking at silver nanoparticles and things like how the silver nanoparticles, either the leaching of the silver into the solution and or the nanoparticle itself, can do things like cause membrane lysing of the antibiotic resistant pathogen. Okay. And so that's, I don't have a ton of experience with that, but that's sort of what I'm limited to. But you can absolutely understand the interaction of those two things and understanding the effect of the pathogen relative to the presence of the carbon dot and how it's affecting the pathogen. Maybe not a complete answer, but it's all I got.
The follow-up question to that question was, if so, can they be employed as reliable diagnostic and or therapeutic agents?
Well, it could be employed as a tool to help you understand the efficacy of your therapeutic agent. Absolutely. You'd have to do the basic and applied research to understand how it would work in that regard. But based on the limited evidence that I've seen, yes, it can. We've even looked at things, for example, as you know, Giardia is a is a waterborne pathogen that can cause major havoc in water drinking systems, potable water. And we looked at therapeutic nanoparticles that absolutely destroy the Giardia pathogen over time. And so that's an example of how it could be utilized. I didn't show any examples of that today. I think we may have some on our website. If not, we can share some. The Giardia just gets destroyed by the silver nanoparticle interaction.
Okay, thank you. Next, how do you dry the samples so that the nanoparticles remain static for the hyperspectral analysis?
Well, that's a good question, and now we're getting in the weeds about sample prep and how it works. And I'm not. Nanoparticles do need to be stationary if they're moving, and in a line scan hyperspectral image capture, they would appear as streaks moving in the sample. They don't have to be dried per se, but they don't need to be in so much solution that they're swimming and or Brownian motion is taking over. You want to have such a small amount or such a large cover glass so that your surface area allows these nanoparticles, even in solution, charge properties to attach to the cover slip or the slide so that you can image them. That's a very nuanced sample prep process that most people have to figure out relative to the details of the chemistry of their sample that they're working on.
Mm-hmm. All right. The next one is from Dimitrios. Good evening, Dr. Cheatham. Given that this microscopy measures scatter, what is the impact of the shape of the nanoparticles in the measurement results? Would deviations from the intended shape, for example, a sphere of the nanoparticles, create a noticeable change in the spectral images?
The answer is absolutely yes. I don't know if you remember the very first methods paper that we demonstrated was a methods paper by Dr. Enrique Toma and his group at University of Sao Paulo. This was a number of years ago, where they looked at different size and different shape plasmonic nanoparticles that produced different optical spectrum that they were able to quantitatively measure. And the other paper that we showed from Dr. T. Pradeep, looking at the ultra small gold nanorod, in that paper, they looked at plasmonic nanoparticles of different shapes and different sizes, and even employed, in that case, cross polarization to look at orientation of these nanoparticles, in addition to their optical spectrum, to understand how different sizes and shapes and deviations change the optical spectrum. Now, simply put, with a plasmonic nanoparticle, a 50 nanometer particle in a refractive index media of water, say, is going to produce a different optical spectrum from 100 nanometer spherical particle, because as surface plasmon resonant based materials, metals get larger at the nanoscale, they're going to redshift, and as they get smaller, they blue shift if they're the same spherical size. For example, a different aspect ratio on a nanorod is going to produce you have a longitudinal and aspect ratio and a transverse spectrum. So it produces two spectrum: the transverse spectrum and the longitudinal spectrum on a plasmonic nanorod. Now, when you start looking at non-plasmonic, not noble metals, that effect can go away, right? I mean, you can infer some things by intensity of signal of metal oxides, for example, but you lose that effect. That works best mostly with metallic-based nanoparticles.
All right, thank you. Next, can we see biocorona clearly around nanoparticles?
As long as there's sufficient biocorona to see, right? If we think about something like polyethylene glycol, PEG, and there's a sufficient molar weight. We've even had groups that are able to look at different molar weights of PEG around the nanoparticle. And that biocorona is shifting the optical spectrum of the nanoparticle. And they can even see different molar weights of PEG that produce different optical spectrum that can be measured. And again, as I mentioned earlier, in the cell, depending on where a nanoparticle is in the cell, it's going to have different biocorona effects. And there are published papers where people have been able to reliably measure that in a way that was quite novel.
Okay, thank you. From Gabriella, how do you prepare slides with live cells? How to avoid medium evaporation?
Yeah, so if you really need to do long-term live cell study, you need an environmental chamber, Gabriella, to do that. And so there are a couple of different environmental chambers that are designed to work with these optics. So you can do long-term studies of live cells if you want to. There's a product by a company called CytoViva that is where they print chips, and they make for very stable cells inside these chips. And you can take them in and out of the incubator. You can image them for 10, 15 minutes, put them back in the incubator. Cells stay stable and healthy in that environment. They can even create these chips so that they can mimic the vasculature nature for a simulated in vivo type of experiment. I'm not an expert on that. The people at CytoViva are. We can provide that environmental chamber as part of a system, or you can acquire it directly from CytoViva. But there are ways to keep your cells happy and healthy so that you can look at them on a time-lapse basis here to look at things like how nanoparticles may be taken up by cells or affected by cells over time.
Wonderful, thank you. Ooh, okay. What are the current technological limitations in hyperspectral microscopy, and what kind of improvements should be addressed in the future?
Wow. I think that there are opportunities, not so much in terms of limitations of the current. Well, there's always a need to create better analysis capability, with all of this data. And so one of the things that CytoViva is always doing is trying to improve the analysis capability. All the analysis capability that we have built in the system today are a direct attribute of customers that say, hey, CytoViva, your hyperspectral imaging is cool, but can it do X from an analysis standpoint? And so over the years, we've added to and enhanced the analysis capability to try to meet those researcher needs as well as possible. There also can be a need, and this is not so much on the research side, but maybe on the clinical or in more material science quality control side, to go really fast, to capture large amounts of data and to analyze that data, let's say in a silicon wafer environment. And so one of the things that CytoViva is looking at are ways to be able to capture spectral data, maybe on a more limited format, a lot faster, and to also be able to analyze that data in real time through all of these really rapidly developing AI algorithms that are out there. So those are some of the things to try to improve on what are some of the limitations, but those limitations are really related to taking it and advancing it beyond most of the basic research that we're doing today, I would say.
Okay, thank you. Next, to collect the spectral library of nanoparticles, is it necessary to manually collect the spectra, or is there an automatic way to do this in the software?
So yeah, you can collect a nanoparticle spectrum, just one spectrum at a time. We have a tool in the software here where we can collect a spectrum and as a large area of spectrum as a region of interest, and then we can convert that region of interest to a spectral library. And then we can take that spectral library and we can filter it against a negative control so that you can be able to have a more automated process for doing that instead of picking spectrum on an individual basis. Sounds like an existing user trying to improve their processes.
All right. Okay, next. Number one, could be any interference with heavy organelles like mitochondria, or maybe with organelles that natively contain metal particles like ferritine.
Absolutely, it could. I mean, if there's ferrous-related materials that are inside an organelle in a cell, it's absolutely possible that that could have an effect on the optical spectrum of the nanoparticle. And that's either good news or bad, right? So if trying to understand if they're ferrous native materials in the organelle, hyperspectral imaging is going to help you identify that potentially. Or if you don't know they're there, it could be a limitation that you'd have to try to work through. So there's not a perfect answer for everything. There's always a if and but to some of these issues that we have to deal with.
All right, thank you. Could CytoViva track nanoparticles inside organoids that have deep thickness?
So we do have some published papers recently that came out looking at organoids and nanoparticle uptake. As I just understood from someone the other day, the FDA here in the US is planning to start accepting organoid spheroid-related data potentially for drug delivery in lieu of in vivo studies, which is very interesting. And yes, there are some papers that if you're interested, we could send you of organoid-related nanoparticle imaging with the tool, where people are trying to get those nanoparticles further and further inside the organoid and with mixed success, I would say, based on some of the data I've seen so far from the CytoViva system. But yeah, it's possible. Again, sometimes sample prep and is very critical to having success, as it is with any microscopy technique that you're looking at. And so sometimes we find ourselves spending as much time working with customers not on how to image or how to analyze, but how to do sample prep to get the data that they really need. And the organoid could be a case of that, for example.
All righty, thank you. 2 more to go.
Mhm.
What type of slides do you use? Is it possible to confuse nanoparticles with particles in dirt slides?
Absolutely, it's possible. Dust particles and nanoparticles, especially those polymer nanoparticles, the dust particles look white, the polymer nanoparticles look white, which is which. So you can use standard off-the-shelf microscope slides from Fisher or whoever for that. Honestly, some of the dirtiest things that you'll ever see in your life, you look at it with CytoViva Darkfield optics or microscope slides. However, we did find a vendor, and Carol, it's Schott, I believe, is the vendor, correct? That
Yes.
produces the ultrasonically clean microscope slides.
Yes.
They're probably triple or more the price of standard slides, but they don't have anything on them. They really are the cleanest thing I've ever seen. And so we recommend, when your experiment calls for having ultra clean slides, so there's no confusion between your nanoparticles and dirt or smuts on the slide, that you use those slides, and we can provide you a link to be able to acquire those if you need to.
Yes, absolutely. I don't have the link readily available, but if you send an email into info@CytoViva, I'll see it, and I can, send you any relative information. Okay, last but not least, is it possible to analyze tissue samples embedded in resin?
It is. The resin, is a mess, quite frankly, from a spectral scattering perspective. But it can be done, and we have done it. And the key to any of this is to have proper controls. To have, for example, I always like to use the analogy that if I'm looking for needles in a haystack, it's always best to start with lots of needles in the haystack, right? My positive control, and to start with a known negative control. And so if I'm looking for something in the tissue and the tissue's embedded in resin, then our nanoparticles, let's look for a heavy dose first. And then the negative control needs to be the tissue and the resin. I will say, though, that if you're using paraffin embedded tissue, if you can deparaffinize, do it. It works a lot better. It just makes it a lot easier. If you can cryosection tissue, that's even better. The less potential interference, the better the images are going to look, the better and easier it is to get to the data. But we can work through the resin soup sometimes, or oftentimes, to do that. Avoid it when possible.
All right, that's perfect. Those were some great questions. Thank you, everyone.
Yep. There was a couple of chats. It said, that's from you. Yep.
That was me. I just told everyone.
All right, gotcha. All right, that's good. Well, with that, we're going to thank you for your time. We went quite a bit over from what we expected, but excellent questions, and we appreciate everybody's time and attention. This is being recorded, and so we're going to allow you guys to be able to have the video of this, so that if you need to share this with colleagues, etc., you can do so. And we hope that everyone has a great weekend, and thank you for your time.
Thank you.